Oh, there you go.
Okay, sorry, we are now recording.
Am I able to start?
Yes, can you share your screen all right?
That's what I'm gonna do.
Okay,
right.
Thanks. All right.
Cool. Let me go ahead and start it.
Yeah, great.
All right, awesome. So thanks for having me. Also, thanks for letting me go first. I like to just get things knocked out. So I'm going to talk about the research I've been conducting with my NIH R15 grant. So I'm entering my third year of funding. So I'm going to talk about risky choice as well as cocaine self-administration. To talk a little bit about risky choice, this involves decisions that involve uncertainty. Not all risky choice is maladaptive. For example, I took a risk by going to University of Kentucky. I grew up in Georgia. I'd never really been to Kentucky, so I had no idea what to expect. So I went off to grad school. That is a risk in itself, but that's totally normal. What I'm more interested in is maladaptive risky choice. For example, you can think of individuals that engage in excessive gambling. So even if they consistently lose money, they keep going back to that behavior. A particular interest to me is substance use disorders. So my background is studying the neurobehavioral mechanisms of addiction, particularly psychostimulant addiction. Drug use in itself is a form of risk-taking. Individuals can overdose and ultimately die. Individuals that engage in drug use can become incarcerated. So if they're using illicit drugs, they can go to prison for that behavior. And so I mentioned I'm interested in neural mechanisms. In particular, I'm interested in the neurotransmitter glutamate. For those that aren't familiar with the neural mechanisms, glutamate is our major excitatory neurotransmitter. There are multiple receptors that glutamate can bind to that exerts effects within the mammalian brain. Specifically, I'm interested in this one receptor. It's called the N-methyl-D aspartate receptor, or the NMDA receptor for short. Essentially, when glutamate is released from this presynaptic neuron, it will bind to NMDA receptors as well as this other receptor over here called an AMPA receptor. And this will allow an influx of positively charged ions like sodium and calcium to flood into the postsynaptic neuron. This is going to cause excitatory postsynaptic potentials, which is a fancy way of saying that it's going to excite this neuron. It's going to increase the likelihood that it sends a message to another neuron. Even more importantly, influx of calcium is going to be important for cellular mechanisms that occur within the cell that can cause changes to this neuron. I'll come back to that later on. If we look at NMDA receptors, they're not all created equally. So the NMDA receptor is composed of 4 subunits that create a channel. When this receptor is stimulated, this channel opens up. That's what allows calcium and sodium to enter the cell. Every NMDA receptor has 2 NR1 subunits. This is where something called glycine binds. There is a combination of glue into A, B, C, and D subunits that can come together. I'm particularly interested in the glue into B, or the NR2B subunit. It's been implicated in substance use disorders. For example, if you target this specific subunit, you can decrease relapse-like behavior in animals. So the point of this grant then is to determine if blocking this particular subunit pharmacologically can decrease risky choice in animals, which is specific aim 1. I was also interested in determining if this drug, which is called RO63-1908, decreases cocaine self-administration in rats. The third specific aim is to look at glue into B receptor binding via receptor autoradiography. I'm currently working on that, so I'm not going to discuss that today. So I'm just going to discuss some of the highlights from specific aims 1 and 2. So how do we measure risky decision-making in animals? There are actually multiple tasks that one can use. For my grant, I'm using a task called the Risky Decision Task. It was developed by an individual named Barry Setlow, who's at the University of Florida. So he's actually a consultant on my current grant. So how this works is an animal is placed into an operant conditioning chamber, which is pictured here on the slide. Animals go through 5 blocks of trials. So they learn initially to press these levers, which you can see down in here. So one lever is going to be associated with a single food pellet. Responding on another lever is going to be associated with a larger payout. So animals can receive twice as much food. However, there's a catch. they may or may not receive a foot shock. So it's a mild aversive stimulus. It's not super pleasurable. So during this task, there are forced choice trials. So that means only one lever comes out. This is to help remind the animal what the contingencies of reinforcement are. So, hey, you're in another block of trials. So now you're going to have an increased chance of getting shocked. Free choice trials just mean that the animal can respond on either lever. So these are the trials that we're ultimately interested in. What do they prefer? So across the session, the probability of receiving a foot shock will increase. So they start each session with a 0% chance of getting shocked. Then they have a 25% chance, 50%, 75%, and 100% chance. So in this experiment, what we did is we determined the effects of this RO63 drug on risky decision-making. So this graph here shows data for male and female rats. To orient you to the data, the x-axis presents the probability of receiving a foot shock. The y-axis is the proportion of responses during those free choice trials for that large risky option. So the large magnitude reinforcer that is paired with a foot shock. What we find is that RO63 decreases risky choice in male rats. So if you look at the red line, this is the highest dose of RO63. You notice that the proportion of responses goes down. So by the time they get to around 75% foot shock, they pretty much refuse to respond on that lever. They instead completely switch their preference to the small, safe reinforcer. What's interesting is that in females, RO63 has no effect. So RO63 decreases risky decision-making in males, but not in females. In addition to examining RO63, we used a compound called CGS19755. This drug is not selective for the gluN2B subunit. Instead, it will target all NMDA receptors. So if it's a gluN2B or a gluN2A or a 2C or a 2D, this receptor will bind, or sorry, this drug will bind to the NMDA receptor and it will block it. So RO63 blocks gluN2B containing NMDA receptors. CGS blocks all NMDA receptors. What's really interesting is that the effects of CGS are completely different compared to RO63. This drug increases risky decision-making in both males and females. And so we were able to publish these data in the journal Psychopharmacology in late 2020. So moving on, I'm going to talk about cocaine self-administration. We use what's called a threshold procedure. How this procedure works is that animals are given 6 6-minute components. So each session is 36 minutes long. They are given, actually they don't get forced choice, that's a mistake. So essentially they just respond to get cocaine infusions. So one little nose poke aperture is associated with cocaine. One is just an inactive lever. So it's going to look something like this. So I have a rat here. So when it sticks its head into that nose poke aperture, it gets an infusion of cocaine. So those lights signal that another response will not lead to reinforcement. This is called a timeout period. This is important just to signal that like, hey, there's no need to respond yet. This is really important for early on in training just to prevent overdose. Cocaine is highly reinforcing in animals. So if you just allow them to respond over and over and over again and get multiple infusions on top of each other, that can be problematic. As they learn this, the timeout period does not need to be as long as they learn to pace themselves. So then with the threshold procedure, what happens is the amount of cocaine that the animal receives decreases across these components. So they start off 0.75 milligrams per kilogram infusion. By the end of the session, they only get 0.002 milligrams per kilogram per infusion, which is a tiny amount of cocaine. So these animals received RO63 injections. Instead of giving them CGS in this experiment, we gave them MK801. MK801, similarly to CGS, blocks all NMDA receptors. It just does it in a slightly different way. To help understand what's going on, the threshold procedure is useful because it allows us to apply behavioral economic analyses. So for example, we can look at what's called initial consumption, which is a proxy for what we call demand intensity. This is the first block of trials. the analogy I like to give to my students is that this would be equivalent to you going to a bar and then a beer is only 50 cents. So in other words, it is consumption when that price is minimally constrained, when it's very low. So animals get a lot of cocaine at that 0.75 milligram per kilogram per infusion block of trials. The other blocks of trials, we can calculate area under the curve. This is a measure of demand elasticity. In other words, how much is the animal willing to respond for cocaine as the price increases? In this case, price increases as the dose of cocaine decreases. They have to respond more and more and more to earn a similar amount of cocaine as they did in the previous block of trials. In humans, this would be analogous to saying, how much are you willing to spend to get a bottle of beer? If it's $5 per beer, are you willing to buy it? If it's $15, are you willing to buy it? What these data show you are the effects of RO63, which is the top row, and the effects of CGS on demand intensity, which is initial consumption, and demand elasticity, which is AUC. For 3 of these panels, you'll notice that there's a single line. So I've collapsed across males and females because there were no differences. With one panel, you'll see a red line and a blue line because there were sex differences. To start with initial consumption, what happens is that RO63 and MK801 decrease initial consumption. Now with MK801, the decrease in initial consumption was more due to a psychomotor impairment. So if you give rats this high dose of MK801, they looked drunk. And so I actually had one student text me and say, hey, can you check out this rat? I think something's wrong with it. And I had to explain to them that that's just normal. Like MK801 just has these psychotomimetic effects. So don't freak out. For demand elasticity, what's interesting is that RO63, even though it decreases initial consumption, tends to increase demand it decreases demand elasticity. So it's going to increase AUC. So they're more willing to respond for cocaine as the price decreases. Wasn't quite statistically significant. However, with MK801, with a dose that does not impair performance, they are much more likely to keep responding for cocaine. So this, in a sense, increases abuse liability in a sense. So that's where we're at. So we're working on these data. So I know I'm running out of time. So I just want to mention that for my competing renewal, I want to try to develop a new risky choice model. The model that I'm using right now has limitations. Because it uses physical pain as a form of punishment, interpreting the effects of glutamatergic drugs can be difficult because those drugs are analgesics. So the question is, if you have a drug like CGS that increases risky choice, is it really increasing risky choice, or is it just making the animal less sensitive to the shock itself? So then another thing, so I met with Bruce and Nigel when they came to NKU last week. And so we talked about looking at cellular mechanisms. So one suggestion that Nigel gave was to look at something called calcium calmodulin dependent protein kinase 2. And so stimulating these NMDA receptors, particularly the gluin 2B subunit, is important for starting this signaling cascade within the cell that can cause these morphological changes. So I thought that was really interesting. So I'd like to incorporate that into my R15. And so with that said, that's pretty much where we're at. That's my update. So any questions? And I'll end my slide. And I will, how do I stop share?
I
Here
think
we go,
you
I see it
just,
right there.
yeah.
There we go.
Whoop, there we go. Okay. Great, thank you. Any questions for Justin?
Justin, thank you for that presentation. So this is a little bit out of my, area of expertise, but I was just curious, what about, how would anxiety play into this? So would RO63 have any impact on, how much anxiety the mice might have when it comes to, anticipating the shock?
Yeah, that's a great point. So I know Barry's lab has looked at the association between anxiety and this risky decision task. They didn't find any correlations. They also looked at shock reactivity. So they look at locomotor activity before. So their operant chambers are set up so they can actually measure locomotor activity. Unfortunately, I can't. So what they find is that locomotor activity doesn't change during that like 1 second shock period. The problem with that though, is that just tells you how well they associate, the shock with their behavior. That doesn't necessarily mean that it's less painful to the animal. So what I really want to try to do is get away from using positive punishment, just because it's really hard to control for that. Another issue, I don't know if I have enough time to get into this. So when you're looking at behavioral pharmacological manipulations, you can control, the shock intensity across animals. So you can titrate it per individual rat. So you can say, hey, this rat needs a higher shock to show sensitivity to the increase of getting shocked. When you're looking at individual differences though, so as a predictor of drug use, what happens is you have to keep the shock intensity consistent. So then you get these animals that never decrease their responding as they continuously get shocked. So it's impossible to interpret those data. Are they high risk taking rats, or are they just merely insensitive to the shock? So because of that, I'm falling out of, I don't particularly like this task. So that's a great question. So yeah, so anxiety, RO63 can have effects on anxiety.
Okay, great. I think we have to move on to our.
Yeah, sorry. All right.
Thanks, Justin.
Thanks, Justin. Yes.
Yeah, no problem. All right, Mark.
Great talk.
So I think I'm next.
Yeah, sorry. I was trying
No
to.
problem. I'll try to keep it brief because I think Bikram and Bradley probably have better things to say than I do. So, but I try not to go too quickly here. So for those of you that don't know who I am, I'm Mark Barget. I'm from the Department of Psychological Sciences at Northern Kentucky University. And so I want to talk about some research I've done over the past, let's see, I guess about 12 or 13 years, basically asking the question, do antipsychotics affect brain development? And most of this talk is going to focus on using rats as an animal model here. So why am I interested in antipsychotics and brain development? So for those of you that know kind of anything or a little bit about psychiatry, antipsychotic drugs are typically used to treat psychosis and schizophrenia, typically in adults. And so, but what's happened over the past couple decades is there's been a pretty dramatic increase in the use of these drugs in children. I think in part because there have been some newer antipsychotics that have been developed that I think some people see as potentially safer, and in some respects they are, but nonetheless, they're still fairly powerful drugs. And so over on the right side of this graph here is actually some homegrown data. So this is data from Kentucky that was developed by Bill Lohr, who is a psychiatrist at the University of Louisville. And he looked at Medicaid records for children that are receiving Medicaid or through their families. And basically what he found was at the beginning of this century, there was a doubling of the use of these drugs in kids from Medicaid families, going from below 0.4% up to 0.8% there, which, if I can round this up a little bit, it's kind of 1 out of every 100 kids on Medicaid are receiving these drugs. And so that's, to me, a pretty significant number of children that receive these medications here. So the most commonly prescribed antipsychotic drug in kids is risperidone. So in Dr. Lohr's graph here, he's showing you with these different bars. The black parts or portion of the bar indicates the proportion of children on risperidone, as opposed to some other antipsychotic drugs that are used. They're most often given to boys relative to girls. And in children, they're most often prescribed when there is a diagnosis in kids with ADHD. Although the FDA has not approved them for that purpose, they have been approved for kids 6 years and up. For kids with autism, risperidone's been approved for them, and aripiprazole or Abilify has also been approved in autism as well. But there are a significant proportion of kids below the age of 6 that also receive these drugs. What's kind of amazing to me, or interesting at least, is that there really are, even to this date, not much data on the long-term effects of these drugs. So basically the question of what happens to a developing brain exposed to antipsychotics in adulthood, what's going on at that point, really hasn't been addressed, and that's been the purpose of my work so far. So how and where do antipsychotics work? And so in terms of how they work, they basically work as neurotransmitter receptor antagonist. So they block principally, in the case of risperidone, dopamine D2 receptors and serotonin 5-HT2A receptors. They also have some affinity for some other serotonin receptors, some receptors for noradrenergic release, and for histamine receptors as well. In terms of where they work in the brain, the principal targets of these drugs have been the dopamine pathways extending from the midbrain, going forward to the forebrain. So one of the main tracks they target looks to be the mesolimbic dopamine tract, where dopamine is produced down here in the ventral tegmental area. It's then shipped up to an area known as the nucleus accumbens, and that tract probably plays a role in the efficacy of these drugs. There's a kind of a branch off this tract known as the mesocortical tract, which may play a role in some of the effects of drugs on memory. There's a separate dopamine tract known as the nigrostriatal system. It works from the substantia nigra. It ships the dopamine up into the caudate putamen, or into the dorsal striatum. And the effects of drugs in that tract probably reflect their motor side effects. So basically, by blocking dopamine receptors in that tract, for instance, they cause some forms of Parkinsonism. So these are the main tracks we're going to look at here, and we're going to focus a little bit on dopamine. I also want to point out, though, that this mesolimbic tract has been implicated in things like reward, just like Justin just talked about, and in cognitive control as well. Again, a topic that Justin kind of touched on there. In terms of how we model this in animals, what we basically do is we take in pregnant dams, we let them have their litters, and from day 14 to 42 of the pup's life, we inject them once a day with either a control substance or risperidone at different doses here. Day 14 is probably analogous to a 3-year-old or a 4-year-old human. Getting out to day 42, we're probably at about a 13 or 14-year-old. We use rats because their brain development kind of tracks along, although at a much quicker pace than what you see in humans. There's a lot known about antipsychotic drugs as well in these animals, and so that's why we've chosen them. Then once we finish the dosing, we give the animals a little bit of time off to let them grow up a little bit, often a week, maybe a month or so, to let them become young adults. And then we start asking them lots of questions behaviorally and biochemically about what's going on in their brains at that point, and maybe how risperidone may have affected that. So one of the first things we noticed about these animals when they are adults is they seem to be kind of more hyperactive when they're in adults. So this is one of the first papers or results we published back in 2013. Julie Franks and Molly Griffith, who were students and staff in my lab, respectively, generated the majority of this data. And basically, we look at animals across days of age from day 49 up until about a years of age, and looking at just their basic activity levels. What we see is in the gray and the black bars, that's the risperidone response versus just vehicles. And you'll notice that these animals are more active pretty much for several months, actually, after we finish giving them the risperidone. So we had another student come along, Bobby Lee Stubman, who did an honors project in my class. Bobby was interested in how these animals respond to psychostimulants that will alter activity as well. And she focused her work, and this was done in collaboration actually with the last speaker, Dr. Yates. And so she focused her work on amphetamine, which is known to be a dopamine, it reverses the transport of dopamine. It basically puts more dopamine out in the synapse, and typically it increases activity in animals. It's used in people in variations as a treatment for ADHD, but it's certainly abused later on as simply a stimulant. And what Bobby looked at here was we took into consideration that our rats with risperidone are already more active. So we kind of did a not really analysis of covariance, but we did a correction to take that difference into account. And what Bobby found was that nonetheless, these animals are still more active when they get amphetamine. So this is an animal that received a high dose of risperidone early in life, looking at 5 hours after amphetamine administration. It's more active than its counterparts down here. But even these lower doses seem to kind of pick up responses. So the animals look more sensitive to the locomotor effects of amphetamine here than what we would normally expect. And then more recently, we've published some data, or I wish we published it. It's actually in review right now, looking at a more direct dopamine receptor agonist. We have somebody with us, it's an expert, Dr. Bruce Mattingly, on the drug apomorphine. And so what apomorphine does is it directly stimulates D1 and D2 receptors. And it has a kind of interesting action when you're just looking at locomotor activity. Often initially, because it blocks D2 receptors on terminal buttons, it shuts off dopamine, and you get animals that are actually less active. So you look at C here, you look at these activity levels, they're decreased for the first few minutes after apomorphine administration, but then afterwards, these drugs start settling on postsynaptic D1 and D2 receptors on the dendrites, and there we see increases in activity. And again, by using the subtraction method where we take off the differences between the risperidone animals and the control animals, we nonetheless see differences here where the animals that receive risperidone, now we're looking at males and females, the risperidone animals are in the dark symbols, the vehicle animals in white symbols. At this lower dose of apomorphine, they're more sensitive to apomorphine in terms of its increase in activity. At a higher dose here, we actually see that the animals on risperidone are more sensitive to the suppressive effects. So it's not just simply activating behavior in general, it's altering behavior according to how the receptor agonist works here. So we've not just looked at locomotor activity, we've also looked at a couple other measures, including sensitivity reward, and also looking at working memory. This work's been done by a former staff member, Tyler Downen, and by two students in my lab, Casey Crane and Emily Baltus-Thompson. Emily did her honors thesis in my lab. And so collectively, these guys worked on an experiment where we asked, do animals that receive risperidone early in life, do they like to hang out in places where they get exposed to drugs of abuse? So in this case, what we did is we gave animals amphetamine. We put them in a particular compartment of a maze, and basically they came to associate that compartment with getting high, if I can put it that way, so that when we put them in this maze with multiple compartments, they tend to hang out in that area where they received their amphetamine before. If you know about this, it's called conditioned place preference. And if we just look at the bars here, so on the y-axis indicates what's the change over time in their preference for this. A positive change here means they like to hang out in this area more. And again, these what we're finding here is the risperidone animals, when they received risperidone when they were young, they tend to hang out more in that area where they got amphetamine before compared to control animals. Our adult animals, we don't see any difference. So the adult group here was actually a group of animals that received risperidone for 28 days or 35 days during adulthood. So just as a control. So it looks like there's a critical period here during development for risperidone to have its effects. What Emily found in a later study was just looking at sucrose reward and how much preference do these animals have for just a bottle of sucrose over a bottle that's hanging right next to it that just has plain tap water in it. And what she found was that in looking at animals during their light cycle and dark cycle, the risperidone animals preferred that sucrose reward more than the vehicle animals did. And then another piece of data here that the three of these folks generated was looking at working memory. So in this case, the animal's in an operant chamber, it's asked to press a lever, and what it has to do is it has to press an opposite lever in order to get a reward. So it has to remember over delay, press this opposite lever, as opposed to some other levers that are present. And so with increasing delays, we see decreases in memory. We found at this early delay, though, that the animals with risperidone didn't do quite so well at remembering what alternate lever to press, are remembering to press that alternate lever. So there's some decreases in working memory going on here as well. So what we found here in terms of looking under the hood and trying to figure out what was going on in the brain is we did find some changes in dopamine transporters and in dopamine receptors themselves. This work was done by two undergraduates in the lab, Matt Gannon and Rachel Stevens, who have since gone on to get their PhDs. And I did this in collaboration with Jim Pauly at the University of Kentucky. The first thing we looked at were dopamine transporters in adult brains that had been exposed to risperidone early in life. And what we found was that the animals that were treated with risperidone had fewer dopamine transporters in their brains. So if you look at these panels over here on the left, this is the frontal cortex. You can follow my arrow, and the lit up areas here in pink are the caudate putamen, anterior caudate, posterior caudate, ventral caudate. What we found in some of those areas, there were decreases in the risperidone animals here in the gray in the caudate in terms of dopamine transporters, which might fit with their enhanced sensitivity to amphetamine, since they're affecting those transporters. Fewer transporters to saturate might be a bigger effect there. And then the other thing we looked at was dopamine D2 receptors. So some of the receptors targeted by apomorphine. And what we found there was the animals with risperidone had higher numbers of these receptors in their frontal cortex and in their anterior caudate compared to vehicle animals. So that may explain a little bit of this increased reaction to apomorphine. So it looks like I've got about a minute left, so I have to apologize. I'm going to skip over our work on neuronal morphology and on microglia and just kind of cut to the chase here. So it looks like early life risperidone increases sensitivity to dopamine and to reward mechanisms. So if this is a seminar about drug abuse, it fits in with that. It also diminishes working memory in adulthood. And some of these changes may be related to some of the stuff I'm reporting here on dopamine transporters and D2 receptors. What I didn't have time to talk about was just some work that we're looking at ascertaining effects on neuronal morphology and on glial cells as well, to get a sense of changes in those areas that we would like to follow up at some point. And so our next steps here would be maybe to go back to our condition place preference studies, look at other drugs that maybe are non-dopaminergic, say opioids, and also then look at the transcriptome of some forebrain regions, looking at neurons versus glia versus astrocytes versus microglia, and seeing how those differ. And so finally, I just want to thank all the students who were involved with the study, the staff who were instrumental in performing them, and a lot of my collaborators as well. So I'll take any questions if there's time.
Great, thank you. Maybe time for one question. It's 4:30 now. We should move on, but anybody have a burning question? Maybe we should just move right on so we stay on track. This is just to try and introduce everybody. Hopefully we'll start conversations. Who is Brad next? Bruce?
Yes.
Yeah. Brad, thanks.
Sorry about that, folks. Couldn't find my unmute button. All right.
Oh,
Going
you
back
were
to-
sharing. Okay.
Okay. Can you all see okay?
Yep.
All right. Thank you, guys. Okay. Thanks, everyone, for having me here today. Due to our 15-minute allotment, I thought I might take this time to talk about sort of a secondary project that I've had going on in my lab related to sort of a new method that we've developed in case any folks in the audience might find this new tool to be useful. And so this relates to a new tool to analyze neurite degeneration in images of cultured cells. So for some brief background, neurite degeneration is this process in which the axons or dendrites that neurons use to communicate progressively deteriorate over time. And it's associated with the early stages of a variety of neurodegenerative disorders, such as the disorders that you see in this list here. But our lab has been particularly interested in the molecular underpinnings of neurite degeneration as it relates to Parkinson's disease. And so about a year and a half ago or so, we became interested in identifying a way to study neurite degeneration using a cell culture model. And so to start off, we chose to use these kind of interesting cells called Lund human mesencephalic cells. And these are cells that, when you thaw a vial of them, they start off looking kind of like your standard neural cell line here. But if you give them kind of a combination of chemicals and growth factors, they differentiate into these really beautiful neurons that have quite extensive neurites. They become electrically active and they form synapses. And so they can be used to model mesencephalic neurons or neurons from the midbrain. And so we first did a variety of tests just to optimize culture conditions, to culture these neurons on microscope slides. Neurons don't like being on glass, and so we had to find some good substrates to get them to stick. And we had to figure out the best density that would give us nice, viable cultures, but yet have individual neurites that we could see, so we could analyze those neurites, without them all being clumped together. Okay, so after we did some experiments and got our culture conditions optimized, in order to induce neurite degeneration, we chose to expose those neurons to 6-hydroxydopamine, which is a neurotoxin that promotes oxidative stress in the neurites. And so you can see here, as we expose these cells to various concentrations of 6-OHDA, the higher the concentration of this drug, the more the neurites become fragmented. And so once we had this sort of model in place for neurite degeneration associated with Parkinson's disease, then the question became, okay, how do we, in a quantitative way, actually measure levels of neurite degeneration? So we can then manipulate variables and see how those variables change the neurite degeneration. And so we chose to use this really commonly cited method that's been used by a diversity of research groups to calculate what's called a degeneration index. And so the way that this works is you essentially take a picture of your cultured neurons through what's called you take a phase contrast image, and then using the open source software ImageJ, also known as Fiji, you binarize that image, so you get black neurons on a white background. And then if there's any cell bodies in that image, you may digitally remove those cell bodies. So you have now just a nice image with only neurites, only axons and dendrites in it. And then ImageJ has this algorithm called, it's a plugin called the particle analyzer, and you can input parameters for size and circularity or linearity. And so by kind of inputting those parameters, you can get that particle analyzer algorithm to detect these little fragments. And so that's what we see here in red. And so the way that one calculates a degeneration index is to add up the areas of all the fragments and then divide that by the total area of all the neurites in the image. And so another way to say it is that the degeneration index is sort of it's reflective of the proportion of neurites that are fragmented. Okay, so we tried using this widely cited method, which I'll now start to refer to as just the traditional DI analysis method, on our cells that had been exposed to 6-OHDA. And you can see that that using this traditional method, we do at least see that with increasing doses of 6-OHDA, we get an increasing degeneration index. We get more neurite degeneration. So far, so good. However, while we were trying out this method, we realized there's actually quite a few different issues. And so we set out to try to improve upon some of these issues. So the first relates to what I call artificial fragmentation. And so this is this phenomenon in which we would have, and it happens quite frequently, you have an image that with neurons that have apparently very intact neurites, but then upon binarization of that image, those intact neurites no longer look intact, they look fragmented. And so we did some different methods of enhancing the contrast of our phase contrast images to see if this was just sort of a issue with kind of needing to process the images a little bit more to boost the contrast. And while qualitatively that helped a little bit, we did some quantitative analyses and found that this artificial fragmentation wasn't really affected too much by just a little bit of contrast enhancement alone. And so then we asked whether maybe instead of taking phase contrast images,
is not available. Record your message at the tone.
I'm sorry, hearing someone's voicemail. So we asked whether using fluorescence micrographs of neurons stained with a cytoskeletal marker may be due to the superior signal to noise ratio of immunofluorescence, whether that would provide enough contrast to really reduce the artificial fragmentation. And indeed, that's what we see. We see the significant reduction in artificial fragmentation when fluorescence micrographs are used. Okay, so another issue that we've noticed with this widely used technique relates to the accuracy of detecting these neurite fragments. And so what we can see here in this image, it highlighted in the blue regions, are all these little fragments of neurites that are going undetected when the conventional parameters that are commonly used for the particle analyzer algorithm are utilized. And so we set out to determine whether we could alter the parameters that are used to more sensitively detect the neurite fragments. And I won't, due to time constraints, I won't go into full details about everything that we changed, but we optimized the parameters to enhance the sensitivity of detection of these fragments. And then we also added in an additional plugin that removes very small non-neurite objects from images to clean up the images and increase the accuracy of our fragment detection further. Okay, another issue that we've noticed related to this common technique relates to the applicability of the technique and its time efficiency. And so most investigators that have done degeneration index measurement, the most common use has been with dorsal root ganglia explant cultures. And they're really amenable for these types of analyses because you have all these cell bodies located in this one cluster. And so you can easily either just physically excise that cluster or digitally, you can remove it. And now you have a nice image with only neurites, right? But a problem is, with dissociated cultures, which are much more commonly employed, if you want to get an image that contains only neurites, the only way to really do that is to digitally kind of use a tracing tool and tediously go around each cell body and digitally remove it. And so what you could imagine is if you have a very large image, or if you're doing, if we extrapolate this over very large data sets with lots of images, that this can make this technique very time consuming and reduces the feasibility of using this type of analysis method for dissociated cultures with mosaically distributed cell bodies. So to solve this, we set out to see if we could somehow automate this whole method, incorporating the previous optimizations that I mentioned, along with somehow adding in automated cell body removal. And so we developed a script for image J that we call ANDI, stands for Automated Neurite Degeneration Index. And so here's sort of what it does. So if one has images of neurons stained with a cytoskeletal marker, like stained for beta-3 tubulin, and then a corresponding image of the nuclei, maybe using a nuclear stain such as DAPI or Hooks. So what Andy will do is take the DAPI image and binarize it, and then it will subject those binarized nuclei to some enlargements to make the nuclei, the binarized nuclei, now encompass about the area of a typical cell body. Then that binarized image will be subtracted from the image from the beta-3 tubulin staining, and then that image will get binarized. And so what we get from this process is this nice image of exclusively featuring neurites. All the cell bodies have been removed. And then the remaining steps of Andy will proceed in with those optimizations that I mentioned before, performing the particle analysis, summing the areas, and then calculating the total neurite area and doing the math to give you a final degeneration index score. So after developing this script and going through many iterations of it, our first question is, okay, is this method of automatically removing cell bodies, would this be a valid way to do this? And so what we did is we took a series of images at different stages of degeneration, and we we calculated the degeneration index, keeping all variables the same with the exception of whether we removed the cell bodies by hand or whether we used the operations in Andy that automate the cell body removal. And we asked how well does the data generated from those two different types of methods correlate? And so what we can see there is we've got this really great correlation where the scores generated following automated cell body removal were almost identical to doing it the old-fashioned way by hand. Okay, so then we asked, all right, if we use Andy, which incorporates all these different, enhancements, use of fluorescence microscopy, optimized parameters for neurite fragment detection, removal of non-neurite material from the images, automation of the cell body removal. If we put all of that together, how do the scores that we get when we calculate neurite degeneration compare to just the traditional method that's most commonly used? And you can see that across different doses of 6-OHDA that cause different degrees of degeneration, we see some significant differences. But so the question is, okay, well, these are, we get differences, but are these differences better? Is Andy really giving us more accurate data or just different data, right? And so after thinking about a way to to test this, we ultimately settled on what I like to just call the eye test, right? And so what we did is we had a variety of investigators who were blinded. We had them in a blinded fashion look at these images of neurons that have different degrees of neurite degeneration and subjectively score them from 0 to 1 related to the proportion of neurites that are degenerated. And then we asked, how well does the traditional method for measuring neurite degeneration correlate with the eye test versus Andy scores? And so what we can see is using the conventional method, there is, a reasonable correlation, an R-squared of 0.78. So a reasonable correlation between subjective measures and the quantitative measures. But when we use Andy, this correlation got much better, in fact, significantly better. And so we think that Andy is generating scores that more closely approximate the neurite degeneration that humans report observing when they look at these images. Also, and very importantly, Andy significantly reduces the time it takes to do these analyses. And so this is a measure of the amount of time per image that it takes to measure the neurite degeneration. And so it's about a 24-fold decrease in time to perform these assays using this automated, using automation via the macro. To give us time, maybe if for any questions, I'm going to skip this last slide, but I'll just summarize in that we wanted to show that Andy could be used in an experiment. And so we performed an experiment. We used an inhibitor of a particular protein and showed that by blocking that protein that it had effects on neurite degeneration. And Andy was able to really nicely help us detect that. Okay, so in conclusion, we've developed this automated method that we think has better accuracy and efficiency. Its applicability is better suited for use with dissociated cultures. It uses all free and open source software, and the script will be released to the public in an open source way with publication of a manuscript. And it's really simple to use. So it's going to really increase user friendliness, maybe for those folks that aren't as familiar with ImageJ, but have an interest in doing these types of analyses. We've got a manuscript related to this that we just got reviews back, and now it's going under revision for resubmission. Overall, editors really liked it and reviewers liked it, but they were curious whether this would be applicable with primary cultures. So if any folks in the audience, we know how to culture primary neurons, but we don't have that up and running, and we're interested in trying to turn that paper around quickly. So any folks in the audience regularly employ use of primary cultures, we'd love to collaborate, or I'd love any suggestions for folks to reach out to. And we're also welcome to collaborators for other projects as well. And you can see sort of a list of the different techniques that we regularly use in the lab. And with that, I'll go ahead and take questions. This work was done, of course, with kind of my army of undergraduate trainees, as well as with my little brother. It was kind of fun. I got to collaborate with my little brother on this. He helped with some of the stats. So appreciate the work of everyone involved in funding from CABIN. And with that, I'll go ahead and take any questions.
Thanks, Brad. That was great. And I have a question, but I think we should move on to the last one. Then we can maybe stay on for questions at the end, just to keep on schedule.
Sure.
Because it's, yeah.
No problem.
Yeah. Okay. So Bikram?
Yes, I'm here. Can I share the screen now?
Yes.
Let me share the screen now.
And everybody can use the chat feature if you want to ask a question that we can discuss later.
Okay. Oh. All right. Hi, everyone. I'm Bikram Suvedi, associate, by the way, now. Associate professor, analytical chemistry at Murray State. Can you hear me? Can you hear me there?
Yeah. Yep.
Okay, I kind of changed my title of my talk a little bit. This is a very nice project we have recently doing or kind of done. we found a significant spike on syringe use and the drug consumption during the COVID-19 pandemic. That's I'm going to talk, and some other stuff that we have done so far. I want to thank all of these undergraduate students and another professor faculty here at Murray State collaborating with me for statistics, Dr. Gopal Nath, and of course, Kentucky NMDA for funding all these projects. As we all know, we have been impacted with this COVID-19 one or other way in some extent, and besides all what we lost, we still have the psychological drugs being heavily used in our community, and prescription rate of the scheduled drugs in Kentucky, as you can see this in diagram during COVID-19 pandemic. The hydrocodone, which is the most prescribed scheduled drugs in state of Kentucky, opioid. It was sliding down actually, as you can see from December 2018 to about March 2020, it was going down. There was some exciting news about going it down, but all of a sudden the COVID-19 started, and as it hits to our community, it was significantly increased then after. That's what we saw. And besides that, all these opioids, hydrocodone, oxycodone, tramadol, significantly increased after COVID-19 pandemic. This is about the prescription rate of the drugs. And not only the prescribed drugs, rate of prescription of drugs being increased, but also the overall overdose death during the COVID-19 pandemic was increased. As you can see this diagram, the y-axis is 12-month ending counts of drug overdose death. This is the data from National Center of Health Statistics, CDC. As you can see, right after that COVID-19 pandemic, as you can see that opioid, and especially opioids, synthetic opioids, and even psychostimulants increased. There was a significant increase on the US overdose death due to these different types of drugs. And of course, Kentucky was not alone. Kentucky overdose death, as you can see in this diagram below, the level of increase in overdose death due to opioid and synthetic opioid was even higher than the national averages. So I want to talk a little bit about what we do in my lab with all this background. We call it wastewater-based epidemiology. And my lab is analytical chemistry lab. We do develop several different types of analytical method capable of determining very trace level of these drug residues in wastewater. The centralized wastewater facility we have in almost all this population community, more than 5,000 in the United States. We collect the wastewater at those sewer treatment plants by utilizing a special auto sampler that withdraw samples like every 15 minutes, for 24 hours to make a really representative composite sample. And we take that sample to the lab, do a lot of sample preparations such as centrifuge, vacuum filtration, special vacuum filtration, and we spike with isotopically labeled drugs of our interest. What we call internal standards. We follow the solid phase extractor, pretty time consuming, exhaustive solid phase extractor. Extraction we do, we concentrate down that volume of about 100 ml to final 1 ml using solvent evaporator, because the level of drugs, as you can imagine, that diluted wastewater is pretty low, so we have to concentrate it by using solvent evaporator. And then we have this UPLC mass spec tandem mass spectrometer, which is, we feel very lucky to have this instrument. And more than that, I'm the only one to use at department. I'm so happy with it. So another small project, well, very important project that we have been doing since last November is under a community where we have this syringe service program. We call it SSP. If you don't know about this program, this is a CDC funded program, which actually targeted to reduce this transmissible disease, such as HIV, hepatitis, by providing the syringe needed for the community. This is, as you can see in this diagram, it's about 220 US counties at risk in the United States. And out of those 220 counties, unfortunately, 54 counties lies within the state of Kentucky. And this red asterisk shows the syringe service program. That's what we have in this community. And the blue shows risk communities. We have so many risk communities, and out of those, 54 communities have this syringe service program. This is a pretty unfortunate that we have this much problem of this transmissible disease and potential overdose death and the drug abuse problems in our state. And as you see in this diagram, what we found was one of the community we have been targeted on this among these 54 counties in the state of Kentucky reported that the syringe dispense has been extremely elevated. As you can see right there at March 20, it has been extremely elevated during the COVID-19 pandemic. Then we thought, why not we monitor the drug residues on the centralized wastewater that we collect on that community. So that's how this project is started. And this diagram shows you the syringe service survey. They also do a survey for the first-time syringe users. They come to the health department and get a syringe, and they also complete the survey. So what we see is this blue line, as you can see, that's a meth, how many doses of meth they use every day, every week. That's the type of questions they ask on the survey, and the cumulative reported dose from that syringe survey, as you can see right from that, the slope of that, I mean, of course it is cumulative reported dose, but as you can see, the slope right after that March 20 is extremely elevated during COVID-19 pandemic. With that all in mind, we, if you want to see how much methamphetamine consumption we have found in that community, it's kind of surprising. As you can see in this y-axis is average consumption rate, milligram per day per 1,000 people. And in the y-axis, different communities. On Australia community, it's about less than 1,000 milligram per day per 1,000 people. There's a couple of European studies we combine. We take an average of a couple of European studies. It's less than 2,000 milligram per day per 1,000 people consumption. Is exactly similar method. That's what we have doing in our lab. That's how this study we're reporting. And a couple of previous our study and the similar study in different part of the United States, east, west, south, north is all over. And this is the data we found is about 7,000 milligram per day per 1,000 people. That's a huge level of drug methamphetamine consumption in the United States. And surprisingly, the community that we have been monitoring is even higher. So the level of methamphetamine consumption, that's what we found in SSP community, is one of the highest level of methamphetamine consumption ever reported. That's what we found. And we continuously monitoring. Is only 2 samples every week that we have been collecting and analyzing since November 2020, and it's still going on process. It's still going on, this project is still going on. And as you can see, this is a trend of the methamphetamine level we found. The consumption rate again, in terms of milligram per day per 1,000 people in y-axis, and the continuous black line is a average running average, and this gray area is 95% confidence interval. That's what we found. So basically, as you can see, during that November, there's couple of red dirts towards the December and the end of December and the first week of January. We found the very high use of methamphetamine during that time. Other than that, there's a regular trend in about 2-3 months. It goes up and it goes down. And that's the type of order we found for the use of methamphetamine in this community. And we tried to correlate the consumption rate, that's what we found, through wastewater analysis versus the reported methamphetamine dose at the health department. And the correlation was not, there was no correlation between that. But however, if we take that November and the December data, which is typically, as we found before, as I described before, was kind of festival time, and if we take those 2 data points out, 2-month data points out, then there's a good correlation we found between the consumption rate, that's what we found in wastewater, versus the reported methamphetamine doses during the syringe collection. Similarly, we also correlated the consumption rate with the number of syringes exchanged in that community, and we found low correlation, but when we exclude again that November and December 2 sort of outlier data, then we found a very good correlation between the consumption rate of methamphetamine with the number of syringes exchanged. So the more the number of people coming to the health department to get syringes, we found the higher the level of methamphetamine in wastewater. What that means is majority of the syringes they've been taking are being used for the methamphetamine consumption. So in another project, we actually also monitor one community in Northwest Tennessee and one community in West Kentucky to look at the isoproteins. If you don't know, isoproteins are the oxidative stress biomarker. We consider them as the oxidative stress biomarker. As somebody is in some psychological stress, it produces a lot of reactive oxygen species that finally produce these isoproteins. And we were monitoring this isoproteins level in wastewater from early 4 months of COVID pandemic, which starts from March 27 to July 1st. We have seen a significant increase in the level of this oxidative stress biomarker. That's what we monitor. And similarly, in another published paper we recently published, we found significantly increased level of hydrocodone consumption. As you can see the diagram at the bottom, in early 4 months of COVID pandemic, from March to June, there was a significant increase in hydrocodone consumption. We found about 72% increase in hydrocodone consumption. And we also try to correlate our data, our findings with other statewide available data. Statewide opioid overdose death or the emergency visit, 50% increase in Kentucky, 100% increase in Tennessee. And that also corroborates with some of the data that's what we have here, is significantly increased, more than 72% increase in the hydrocodone consumption we found. Similarly, we also tried and we hypothesized that the hydrocodone, I don't know whether you remember, but during that COVID-19 pandemic, CDC and SHAMS actually allowed the prescription of hydrocodone through the tele-prescription. People could not go to the hospital, they could call pharmacy and get the hydrocodone prescription. That is potentially a reason why we had this steady increase in hydrocodone besides other things. And overall, statewide opioid citation trafficking was increased by 47% that time, and that also tells are aligned with the data that we found. Conversely, the psychostimulants such as methamphetamine and cocaine, of course, they were not prescribed drugs. And the use of these drugs, because of all these international and national traffics are down, potentially some of these drugs were not available in community. As a result, there was a small increase from March to April. As you can see on the top diagram, in one community A, there was a significant increase of the use of methamphetamine from March to April. Then after that, it was continuously going down. And as the pandemic starts the business starts reopening slowly, then at that time, the level of that methamphetamine consumption was again going up. So this shows that it's also the availability of these drugs in community also potentially have impacted the use profile of these drugs. So we also try to correlate some of our findings with the statewide findings, conventional way of findings. The psychostimulus possession citation, meth was decreased 12.6%, cocaine was 54%, and that also those information was also aligned with the data that's already found. And even CD police, the possession citation of the meth was decreased by 43%, and similar data what we found. And as I said before, the limited supply and the availability could be related to the increased demand and the travel restriction, as well as the drug price increase, the hike in a drug price. And so this data has already been reported, has already been published. These are the papers we have been able to publish due to Kevin funding. And all of these papers are actually the undergraduate researchers are the lead co-authors or the lead authors or co-authors, and some faculty here at Murray State and also University of Louisville. This Kevin grant was supporting 12 undergraduate researchers for the past 4 years, and we had more than 20 scientific presentations from this. And I don't know this is the part of this presentation or not, but I have an update on R15. 2017, 2018 were not discussed. I submitted R15, it was not discussed. In 2019, it's barely scored. I got 45. And recently, well, in the month of June, I submitted again, June or July. Yeah, I submitted that R15 in 2021 again. It's a comprehensive wastewater epidemiology assessment of opioids in healing communities in Kentucky. We name it We Heal KY, Kentucky. And I'm really excited about this, but I'm just waiting to hear back from them. So I want to acknowledge all of my undergraduate researchers and Dr. Gopal Nath for contributing on all these projects, and Jones Research Center here at Chemistry Department for providing access to the instrument, and also, and of course, Kentucky Navy for providing all these fund and making all this research possible. And I would be more than happy to answer if any question I can.
Great, thank you, Bikram. I think, we're a little over, but we can continue discussing if people can stay on for a little bit. And, just you can ask questions of any of the presenters. I was just curious, Bikram, I know some groups are looking at, different COVID variants. I don't know if they can detect amount from wastewater. I mean, they're looking at COVID variants in wastewater. Is there any, do you know if they can detect the amount, and could that be correlated with your data? Or?
Well, you know what, we also did that for the past, I think we started on November 2020 and we did until April 2021. For the 6 months, we actually collected wastewater sample from the same community. That's what I mentioned, that SSP community. And looked over the COVID variant, and we found a very detectable level of COVID variant, and we correlated that with the COVID cases that daily reported by the health department, and we found a very exciting correlation at that time. And that information actually used by the health department of that particular county, let me not disclose, but that county utilized all those information along with our drug data, and yeah, I think I wish we could continue that, but I didn't have enough funding to continue that project, and we stopped. But it was an exciting project.
Yeah, that's neat. There's so much data you can get from wastewater.
Exactly.
I have a question. So, Brad, are you still on?
Yes.
Yeah, I was just curious. Again, in your method, that was really cool. The antibody that you used to detect the dendrites, what antibody was that?
So that was just a beta-3 tubulin antibody. So
Oh,
it would detect axons and dendrites. But yeah, it's a real nice beta-3 tubulin antibody from Covance. Okay,
I was just curious, does the type of antibody you use affect how many of the fragments you might detect? Like if the beta tubulin is degenerating too, is there more fragments maybe than you're detecting? I don't know.
Yeah, that's a great question. And it's one of the things when we first set out to do it that we were really curious about is, would the way that beta-3 tubulin levels change actually reflect the changes in the total axoplasm or the total cytoplasm of the axon. But at least in terms of those, those correlations that we did. So when we had blinded investigators subjectively measure the degeneration, they were doing that with phase contrast images. And so what we found is that with the beta-3 tubulin staining, with the Andy macro, we were still getting better scores. Those scores were matching up better with subjective scoring of phase contrast images than actually quantifying the phase contrast images with sort of the conventional method.
That's cool. Have you looked at phase contrast? Like you're looking at a certain image, here's phase contrast, here's fluorescence. You basically see the same, amount of dendrites, et cetera. Or
yes,
do you think your
I
phase
see what you're
contrast
saying.
would detect
They match up really, like qualitatively, they match up really well. With phase contrast, you get a few more of those kind of small, like just little debris you see better. And so that's almost an advantage of beta-3 tubulin is that, you're less likely to see just like
dirt?
yeah,
or
exactly.
whatever.
Random gunk in the extracellular space. So yeah, that's I think a benefit of fluorescence microscopy as well.
Okay, cool.
Thanks.
Mark, I wanted to see your morphology stuff. You skipped
I'm sorry,
over.
I practiced that talk and we all came in at 10 minutes when I practiced it.
I know. I
So.
think we're realizing that the 15 minutes is a little short, but we thought for an initial.
No, it's my bad.
Yeah.
But I mean, basically, the real quick answer is there is not a lot of changes there. I think it's still very preliminary, looking at morphology, looking at spines, and then looking at microglia. So
in the animals that have risperidone
yes,
versus
yeah, right.
So Mark, on those, the different effects you were seeing in adulthood with regard to sensitivity to drugs or other effects or activity, but they look like with continued testing out, they eventually disappeared. Is that right?
Yeah. So that very first study we did, it looked like about a year out, it went away. So
do you know?
Although,
go ahead.
can I just real quickly? That study just looked at male rats, and we're seeing much bigger effects in the female rats. So be interesting to go back and look at that with this was pre, before you had to have NIH requested people, and which is good. I mean,
yeah, absolutely. So, but I would expect you to be following the clinical literature that close, but these kids, they get these antipsychotic drugs, do they ultimately stay on them through adulthood or just for a few years? Or
I think for the most part, they're used temporarily. So I think when an incident happens or there's a problem, kid stays, I think they're, the mean time is somewhere between like 6 months to a year or so. But I know that, anecdotally, there are kids that stay on them for a very long time into adulthood. So I think I'm more focused on what happens to the kids that get them is not maybe a quick shot, but a little bit of exposure at a critical period of what happens later on.
No, important.
Yep.
Well, any other questions? I think that there's a lot of overlapping interests. I think hopefully you guys can continue to discuss on your own.
It's really good.
No further questions. Great presentations. I really enjoyed it, and see you all in 2 weeks.
Thanks, everybody. Bye.
Bye.
Stop.